Welcome to the Deep Dives, where we take complex information and plunge into the fascinating insights hidden within. Today, we're embarking on a journey into the incredible inner world of your alimentary tract.
That's right. We've gathered source material that really lets us explore its fundamental functions, how food actually moves through you, how this whole intricate process is controlled by its very own nervous system, and this whole symphony of hormones.
And critically, how it gets the vital blood supply it needs to actually do its job. It's easy to forget that part.
Absolutely. Think of this deep dive as your essential guide to understanding the sophisticated machinery of digestion and absorption, or really trying to move beyond sort of the basic diagrams you might remember.
Yeah, the high school biology stuff.
Exactly. To truly appreciate the remarkable physiology that is constantly, tirelessly working within you, it's quite amazing when you get into it.
It really is. Our mission today is to unpack this material together, pulling out the absolute key nuggets of knowledge and connecting the dots to reveal the sheer mastery of this system. You might think you know your gut, but trust us, there's a universe of complexity in there.
Definitely.
Okay, let's unpack this.
So, starting with the big picture, the alimentary tract at its core has one overarching, absolutely essential role: providing the body with a continuous, reliable supply of water, electrolytes, vitamins, and nutrients.
It's the fuel line, basically.
Look, it's the fuel line, the water line, the building block supplier. It's the primary way your body gets everything it needs to survive and function. And importantly, it's not just a passive pipe. It's an active processor and delivery system.
Right. And to actually fulfill that role, that continuous supply mission, our source material breaks down the process into 5 fundamental functions. These are absolutely necessary.
Yeah, it's 5 key things that have to happen, often simultaneously.
What are they?
First is the movement of food, propulsion. Food has to get from your mouth all the way through, eventually, to the anus. Sounds simple, but controlling that movement at just the right pace through all the different specialized segments, that's incredibly complex.
Because it's too fast?
Right. Too fast, you don't absorb enough nutrients. Too slow, things get backed up. You get blockages, discomfort. Timing is everything.
Okay, number 2.
Second, you need the secretion of digestive juices. These are things like enzymes, acids, bicarbonate, mucus, produced by glands all along the tract. Their job is crucial for the third function,
Which is digestion. Breaking stuff down.
Exactly. Digestion is breaking down those complex food molecules, the big proteins, fats, carbohydrates, into smaller components that your body can actually handle and absorb. The juices provide the chemical tools for that.
Makes sense. So move it, secrete juices, digest it. What's next?
Once it's broken down into small enough pieces, you need function number 4, absorption. This is where the magic really happens, getting that water, those electrolytes, vitamins, and the small digestive products out of the inside of the gut tube.
Across the gut wall.
Precisely. Across the intestinal wall and into the body's internal environment, usually the bloodstream or the lymphatic system.
And it takes energy, right? And needs transport.
Absolutely.
Yeah.
Which brings us to the 5th essential function, circulation of blood. You need a dedicated, robust blood supply flowing through all these gastrointestinal organs. It fuels the muscle movement, powers the secretion, drives absorption, and then, crucially, carries away all those absorbed substances to the rest of the body. It's the delivery network. It's the delivery network and the power supply. It ensures the gut wall itself gets the oxygen and energy it needs to perform all these demanding tasks.
And looking at the whole system, like the diagram in the source shows, mouth to anus. It's amazing how each part is so different.
Oh, completely. Each distinct part, the esophagus, stomach, small intestine, large intestine, is specifically adapted structurally and functionally for its particular role within these 5 processes.
Like the esophagus is just a tube, basically.
Primarily a passageway, yeah, designed for rapid transit. Then the stomach, it's this muscular bag built for storage, mixing, and starting that initial protein and fat breakdown with acid and enzymes.
But the small intestine, that's the main event for digestion and absorption.
That's where the vast majority happens. And its structure reflects that incredible length, folded lining, tiny projections called villi, all creating a massive surface area for maximum absorption. Then the large intestine handles mostly water, electrolytes, and waste storage. It's a real masterclass in form-following function.
So to really get how the gut does all this, especially the movement part, we need to zoom right in on the wall itself. The structure.
Exactly.
Yeah.
The source gives us a great typical cross-section. Imagine looking at a slice of the intestinal tube.
Like layers of an onion.
Sort of, yeah. From the very outside, you've got this thin protective layer called the serosa.
Oh.
Just inside that are the real workhorses for movement, two thick layers of smooth muscle. The outer one is the longitudinal smooth muscle layer where the fibers run along the length of the gut.
And inside that?
Inside that is the circular smooth muscle layer where the fibers wrap around the gut's circumference.
So one squeezes lengthwise, one squeezes diameter-wise.
Yeah.
Makes sense.
Precisely. These two layers are absolutely key for the mechanical stuff, squeezing, churning, pushing food along. They generate the force.
Got it. What's underneath the muscle?
Beneath those muscle layers is the submucosa. This is a layer of connective tissue, but it's packed with important stuff. Blood vessels, lymphatic vessels, and crucially, a part of the gut's own nervous system, which we'll definitely get back to.
Okay, the plumbing and wiring.
Pretty much. And then the innermost layer, the one actually lining the lumen where the food is the mucosa. This is your absorption and secretion specialist layer.
And does it have muscle too?
It does, interestingly. A very thin inner layer of muscle fibers called the mucosal muscle, or muscularis mucosae. It's much sparser than the main layers, but it can cause folds and ridges in the mucosal lining.
So lots of layers, each with a job. But for the big movements, we're looking at those main longitudinal and circular smooth muscle layers.
Absolutely. They are the engines of motility.
And how does that muscle actually work? Is it like skeletal muscle?
Similar in principle, but quite different in detail. If you look really close, the individual smooth muscle fibers are tiny, maybe 200 to 500 micrometers long, only 2 to 10 micrometers thick.
Really small.
Very. And they're arranged in bundles, sometimes up to 1,000 parallel fibers running together within one bundle. In the longitudinal layer, the bundles run lengthwise. In the circular layer, they run around.
Okay, bundles of fibers, how do they coordinate?
Now this is where it gets really interesting and fundamental to how the gut works. The fibers within a bundle aren't electrically isolated islands. They're connected by specialized structures called gap junctions.
Gap junctions, I've heard of those, like little tunnels.
Exactly like little tunnels or electrical windows between adjacent cells within the same bundle. They let ions, those charged particles that carry electrical signals, move directly from one cell cytoplasm to the next with very, very little resistance.
So if one cell gets an electrical signal,
that signal can spread rapidly and easily to all its connected neighbors within that bundle. It travels particularly fast along the length of the bundle, following the orientation of the fibers.
okay, so fibers in a bundle act together. But what about different bundles?
Good question. The source points out that the bundles aren't completely separate either. They actually fuse together at many points throughout the muscle layer. There are connections between bundles, too.
So it's all interconnected.
It's highly interconnected. And this widespread electrical connection both within bundles via gap junctions and between bundles where they fuse means that each entire muscle layer, longitudinal or circular, doesn't behave like a collection of individual independent muscle cells. It functions as an electrical syncytium.
Syncytium. Okay, that sounds important. What does it mean practically?
It means that when any one part of that muscle layer gets electrically excited, receives an action potential, that electrical signal doesn't just stay localized. It generally travels like ripples in a pond in all directions throughout that entire muscle layer.
Wow. So stimulating one spot can affect a whole area.
Exactly. The actual distance the action potential travels depends on how excitable the muscle is at that moment. It might only go a few millimeters if excitability is low, or it could propagate for many centimeters, coordinating a contraction across a significant length or circumference of the gut if excitability is high.
And do the two layers, longitudinal and circular, talk to each other electrically too?
Yes, there are also connections, gap junctions, and fusion points between the longitudinal and circular layers. So excitation starting in one layer can, and often does, spread to the other layer, allowing for highly coordinated multi-directional contractions. This syncytial property is absolutely fundamental to how the gut generates widespread coordinated movements like peristalsis.
Okay, that syncytium idea is key. It explains how a large section can contract together. Now, what about the electrical signals themselves? You mentioned action potentials.
Right. This leads us to the really fascinating electrical life of the GI smooth muscle. It's almost never truly electrically silent. There's almost continual, low-level, intrinsic electrical activity happening.
Like a background hum.
Kind of, yeah. And this activity primarily takes two basic forms, which the source shows conceptually on a graph. There are what we call slow waves, and then often riding on top of those are spike potentials.
Slow waves and spike potentials. Got it.
Plus, there's a third factor, The baseline resting membrane potential, the sort of average electrical charge across the muscle cell membrane when it's resting, isn't absolutely fixed. It can shift up or down, becoming more or less negative. And these shifts are incredibly important because they drastically change how easily the muscle gets excited. They're a major control point for motor activity.
Okay, let's break those down. Slow waves first. What are they exactly? Are they contractions?
No, and this is a critical point the source emphasizes. Slow waves are not action potentials themselves, and they generally don't cause muscle contraction directly.
Okay, so what are they?
They're best described as slow, rhythmic, undulating changes in that resting membrane potential. Think of it like a gentle electrical tide, ebbing and flowing, the potential becoming slightly less negative, then more negative, then less negative again in a regular pattern.
How big are these waves? And how fast?
Their intensity, the swing from trough to peak, is usually around 5 to 15 millivolts. And their frequency, how often they occur, is very characteristic for each part of the gut, but remarkably constant within that part. For example, in the stomach, it's slow, maybe 3 per minute. In the duodenum, the first part of the small intestine, it speeds up to around 12 per minute. Then it slows down a bit towards the end of the small intestine, the terminal ileum, to maybe 8 or 9 per minute.
And why is that frequency important?
It's vital because, as the source makes clear, the slow waves essentially set the rhythm for most gastrointestinal contractions. The gut muscle in a particular segment can generally only contract phasically at the frequency determined by the slow waves in that segment. They are the pacemaker.
What generates them? The muscle cells themselves.
It's a bit more complex. It seems to be an interaction between the smooth muscle cells and a network of specialized cells called the interstitial cells of Cajal, or ICCs.
ICCs. Okay, what do they do?
These ICCs are thought to be the primary electrical pacemakers. They form these intricate networks, especially located between the muscle layers, and they make very close, almost synapse-like contacts with the smooth muscle cells.
So they generate the rhythm.
That's the leading theory. The idea is that ICCs have unique ion channels that open and close cyclically, generating rhythmic inward electrical currents. These currents then spread through those gap junctions we talked about into the smooth muscle cells, creating the undulating slow wave pattern in the muscle membrane potential.
Fascinating. But you said they don't usually cause contraction?
Correct. By themselves, slow waves usually don't trigger a significant contraction, maybe a tiny bit in the stomach, but generally no. Their main job is to control the timing and pattern of the other type of electrical activity. The spike potentials.
The spike potentials.
Yeah.
These are the action potentials.
Yes, spike potentials are the true action potentials in GI smooth muscle. These are the sharp, rapid electrical depolarization events that do directly trigger significant muscle contraction.
And do they happen?
They fire automatically whenever the resting membrane potential of the smooth muscle cell depolarizes, becomes less negative enough to reach a critical threshold, which is typically around negative 40 millivolts. Remember, the normal resting potential is more negative, maybe negative 50 to negative 60 millivolts.
So the slow wave comes along.
And its peak, that most depolarized point, rises high enough to cross that negative 40 millivolt threshold. Bang, you get one or more spike potentials firing rapidly right there on the peak of the slow wave.
I see. So the slow wave brings the potential up towards the threshold.
Exactly. And if it reaches it, the spikes fire. And the higher the slow wave peak goes above that threshold, the more frequently the spike potentials will fire during that peak. It might be just one spike, or it could be a burst of 5 or 10 spikes per second.
So more spikes means stronger contraction.
Generally, yes. The frequency of spike potentials correlates with the strength of the muscle contraction. The slow wave sets the timing window, and the spike frequency within that window determines the intensity.
How do these spikes compare to nerve action potentials?
They're quite different in duration. The source highlights that GI muscle spikes are much, much longer, typically lasting 10 to 20 milliseconds, which is 10 to 40 times longer than a typical nerve action potential.
Why so long?
It's down to the type of ion channels involved. Nerve action potentials are mainly driven by the super-fast influx of sodium ions through voltage-gated sodium channels.
But in the gut muscle?
In GI smooth muscle, the channels responsible for the spike are different. They're called calcium-sodium channels. These channels are much slower to open and slower to close compared to the nerve's sodium channels. And critically, when they open, they allow the entry of a large number of calcium ions along with a smaller number of sodium ions.
Calcium. That sounds important for muscle.
Absolutely crucial. We'll come back to that. But the slowness of these calcium-sodium channels is the reason the spike potential lasts so much longer. And the fact that they let in a lot of calcium during that spike is fundamental to triggering the actual contraction.
Okay, so just to recap that sequence.
Right.
Slow waves set the rhythm, and if they depolarize the membrane enough to hit the threshold,
Then spike potentials fire on the peak.
and the spikes are long because of slow calcium-sodium channels.
Right.
And it's those spikes letting calcium in that actually cause the contraction.
You've got it. That's the core mechanism for most rhythmic contractions. Slow waves, timing. Spike potentials can trigger and calcium delivery for contraction.
Now, you also mentioned the baseline resting potential can change. How does that fit in?
Right, so besides the waves, the whole baseline level, that average -56 millivolts or so, can shift.
Yeah.
If it becomes less negative, we call that depolarization. The muscle fiber becomes more excitable.
Because it's closer to the -40 threshold.
Exactly. It takes less of a push from the slow wave to trigger those spikes. So depolarization increases the likelihood and frequency of spike potentials, leading to stronger or more frequent contractions.
And the opposite, more negative.
If the baseline becomes more negative hyperpolarization, the fiber becomes less excitable. It's further away from the threshold. The slow waves might not reach it at all or only trigger very few spikes. So hyperpolarization reduces or prevents contraction.
What causes these shifts in the baseline?
Several things can depolarize and excite the muscle. The sourceless stretching of the muscle wall, just the physical presence of food stretching it. Also, acetylcholine, the neurotransmitter released by parasympathetic nerves, and certain specific gastrointestinal hormones, these all tend to make the resting potential less negative.
And what makes it hyperpolarize, more negative, and inhibited?
The main players here are norepinephrine, released by sympathetic nerves, and epinephrine, adrenaline, circulating in the blood. These neurotransmitters make the membrane potential more negative, moving it away from the threshold and thus inhibiting muscle activity.
So stretch, parasympathetic nerves, and some hormones ramp things up by depolarizing. Sympathetic nerves and adrenaline shut things down by hyperpolarizing.
That's a good summary. These baseline shifts are a major way the intensity of gut contractions is controlled, working alongside the rhythm set by the slow waves.
Okay, let's nail down that calcium point. You said its entry causes contraction. How?
Yes, this is fundamental physiology. Calcium ions entering the smooth muscle fiber during those spike potentials are the direct trigger. They bind to a regulatory protein called calmodulin.
Calmodulin, okay.
This calcium-calmodulin complex then activates an enzyme cascade that ultimately allows the myosin heads, the motor proteins, to interact with the actin filaments and pull, generating force. It's the standard final step in muscle contraction, though the initial regulation via calmodulin is typical for smooth muscle.
And just to be crystal clear, slow waves don't do this.
Right. The ion movement during slow waves is mostly sodium, and it doesn't cause a significant enough influx of calcium to activate that calmodulin pathway. That's why slow waves alone generally don't cause contraction.
It's the spike potentials, opening those specific calcium-sodium channels
Yep.
that let in the necessary calcium flood to actually make the muscle contract powerfully.
Precisely. The spikes are the calcium gatekeepers for significant rhythmic contractions.
Now, are all gut contractions rhythmic like this, tied to slow waves and spikes?
Not entirely. The source also describes another type called tonic contraction.
Tonic, meaning constant.
Yeah, essentially. It's a continuous contraction, not rhythmic like the waves. Its intensity can go up or down, but the contraction itself persists for long periods, minutes or even hours. It's more like a sustained clamping or squeezing rather than rhythmic pulsing.
Where do you see that? Like in sphincters?
Sphincters are a prime example, maintaining closure. But it can occur elsewhere, too. The mechanisms causing tonic contraction are actually a bit less clear than for rhythmic contractions.
How might it happen?
Sometimes it seems tonic contraction is related to continuous, repetitive spike potentials firing at a very high frequency. The more frequent the spikes, the stronger the sustained contraction.
But sometimes it happens without spikes.
Yes, it seems it can. Certain hormones or other factors might cause a continuous partial depolarization of the muscle membrane. Maybe it doesn't quite reach the -40 milliV threshold for spiking, but it's enough to allow a slow, steady leak of calcium ions into the cell, leading to a low-level sustained contraction.
Or maybe calcium gets in some other way.
That's also possible. The source mentions that sometimes tonic contraction might involve continuous calcium entry through pathways not directly linked to changes in the membrane potential at all. The bottom line is tonic contraction is important for sustained pressure or closure, but its exact control mechanisms are varied and not fully mapped out in all cases.
Okay, so we have the muscle, its syncytial nature, the electrical rhythm slow ways, setting the pace, spikes triggering contraction via calcium, and baseline shifts controlling excitability, plus this other tonic mode. That's a lot going on just in the muscle itself.
It really is. But how is all this activity orchestrated along the entire 25-plus feet of the gut? How does the stomach coordinate with the intestine? That brings us to the gut's own command center, the astounding enteric nervous system, or ENS.
The second brain. I've heard that term. Is it really that complex?
It truly is. The ENS is an entire self-contained nervous system embedded within the wall of the gut, running all the way from the esophagus down to the anus. It can function with remarkable independence from the actual brain and spinal cord.
And the scale. The source says over 100 million neurons.
That's right. More neurons than you find in the entire spinal cord. It's a massive, intricate network dedicated to running the gut.
What's its main job?
Primarily controlling gastrointestinal movements and secretion. While it definitely communicates with and is influenced by the central nervous system, the ENS handles a huge amount of local control and can execute complex reflexes entirely within the gut wall.
Where is it located within those gut wall layers we discussed?
It's organized into two main interconnected networks, or plexuses. The source shows this clearly.
Okay, two plexus.
The outer one is the myenteric plexus, sometimes called Auerbach's plexus. Myo relates to muscle, and enteric to the intestine fittingly. It's located between the longitudinal and circular smooth muscle layers.
Right in the action zone for movement control.
Exactly. The inner plexus is the submucosal plexus, or Meissner's plexus. As its name suggests, it's located deeper within the submucosal layer, underneath the muscles, but above the innermost lining.
And do they do different things?
They have somewhat distinct primary roles, though they're interconnected. The myenteric plexus, being sandwiched between the muscle layers, is mainly concerned with controlling gastrointestinal movements, coordinating the contractions and relaxation of those muscle layers for propulsion and mixing.
And the submucosal plexus, deeper in.
The submucosal plexus, located closer to the lining, focuses more on regulating local functions within the inner wall of each segment. This mainly involves controlling gastrointestinal secretion from the glands embedded there, and also regulating local blood flow to support secretion and absorption.
So, myenteric for movement, submucosal for secretion and local stuff.
That's the general division of labor, yes. But remember, they talk to each other and work together.
Now, you said the ENS can work independently, but it is connected to the main nervous system.
Absolutely. While the ENS has autonomy, its activity is heavily modulated by input from the extrinsic autonomic nervous system, the sympathetic and parasympathetic systems originating outside the gut. Fibers from both systems connect extensively to neurons in both the myenteric and submucosal plexuses.
So the brain and spinal cord can override or adjust what the gut brain is doing.
They can greatly enhance or inhibit its functions, yes. They provide that higher level of coordination with the rest of the body's needs.
And how does the ENS know what's going on inside the gut? Does it have sensors?
It does extensively. The gut wall is packed with sensory nerve endings. These receptors are found in the epithelium lining the lumen, detecting chemicals and irritation, and also within the deeper layers of the gut wall, detecting stretch or distension.
So they're constantly monitoring the internal environment.
Constantly. And these sensory nerves send signals, afferent signals, carrying information away from the gut. Some signals go just to other neurons within the ENS plexuses, triggering purely local reflexes.
But others go further.
Yes, other sensory signals travel outwards. Some go to the prevertebral sympathetic ganglia located outside the gut. And many travel all the way up to the spinal cord and even the brainstem, especially via those large vagus nerves.
So the gut is constantly sending feedback upstairs.
It's a crucial feedback loop. It informs the ENS and the central nervous system about the state of affairs. Is there food? Is it acidic? Is the gut stretched? Is there irritation? Allowing for appropriate and coordinated responses from simple local tweaks to complex body-wide reflexes.
Let's dig into those plexuses a bit more. The myenteric plexus, between the muscle layers, what's its structure like?
The source describes it as forming a more or less continuous linear chain of many interconnected neurons that extends the entire length of the GI tract.
A long network.
Exactly. This structure, running lengthwise and connecting neurons over distance, is perfectly suited for its main job, controlling muscle activity along the length of the gut, coordinating those propagating waves of contraction and relaxation needed for propulsion.
What happens if you stimulate it?
Generally, stimulating the myenteric plexus ramps up muscle activity. The source lists several principal effects. Increased tonic contraction, so the gut wall has more tone. Increased intensity of the rhythmic contractions we talked about, likely by making it easier for spike potentials to fire. Possibly a slight increase in the rate of the slow wave rhythm. And very importantly, an increased velocity of conduction of excitatory waves along the gut wall.
Faster conduction means faster peristalsis.
Precisely. It allows those propulsive waves to sweep along the gut more quickly and effectively, moving contents along efficiently.
Is it always excitatory, though? Does it ever tell the muscle to relax?
That's a key point the source makes. No, it's not purely excitatory. The myenteric plexus also contains numerous inhibitory neurons.
Inhibitory neurons, what do they do?
They release inhibitory neurotransmitters. The source mentions possibilities like vasoactive intestinal polypeptide, VIP, or others which act to suppress muscle activity in specific locations or at specific times.
Why would you want to inhibit muscle contraction?
Think about those muscular rings, the sphincters, that act like valves between different sections of the gut, like the pyloric sphincter controlling stomach emptying, or the ileocecal valve between the small and large intestine. These need to be tightly closed most of the time to regulate the flow of contents.
Ah, so the inhibitory neurons tell them when to relax and open.
Exactly. When a peristaltic wave approaches a sphincter, inhibitory signals from the myenteric plexus cause the sphincter muscle to relax just ahead of the wave, allowing the contents to pass through. Without this crucial inhibitory function, food would just pile up behind closed doors. It's essential for coordinated flow.
Okay, so the myenteric plexus handles the big movements, both contraction and relaxation, along the length. Now, what about the submucosal plexus again? Meissner's plexus in the submucosa.
Right. Its focus is different. The source contrasts it with the myenteric, saying it's primarily concerned with controlling function in the inner wall of each minute segment of the intestine. It's much more localized.
Local control. What kind of local control?
It acts as the integration center for many sensory signals coming directly from the gut lining, the epithelium within that small segment. It senses the chemical nature of the food, the physical presence, the pH, etc., right there.
And based on that local information?
Based on that, it helps control local intestinal secretion from the glands in the mucosa, local absorption processes, and also local contraction of that thin mucosal muscle layer we mentioned earlier, which can change the shape of the inner lining, maybe creating folds to increase surface area or help stir the contents right near the surface.
So myenteric long-distance movement coordination, submucosal elective, local fine-tuning of secretion, absorption, and the inner wall shape within each segment.
That's a great way to put it. They operate at different scales but work together as part of the overall ENS control system.
And the communication within this system.
Yeah.
You mentioned neurotransmitters. How many are there?
It's astonishingly complex. The source mentions that over 25 potential neurotransmitter substances have been identified as being secreted by various enteric neurons.
25. That's way more than just on-off signals.
Absolutely. It gives you a sense of the incredible nuance and sophistication of the chemical signaling happening entirely within the gut wall. It allows for very specific and graded responses.
Are there any main players we should know?
The source highlights a couple with well-established general effects. Acetylcholine is probably the most common excitatory neurotransmitter released by ENS neurons. It generally stimulates or enhances GI activity, more contraction, more secretion.
And the main inhibitory one?
That would be norepinephrine. It's primarily released by the extrinsic sympathetic nerves that connect to the ENS, but some intrinsic neurons might use it too. Norepinephrine almost always inhibits GI activity. Epinephrine, which is adrenaline from the adrenal glands circulating in the blood, also has a similarly inhibitory effect.
What about all the others listed? Serotonin, dopamine, CCK, VIP, substance P.
That's where it gets really detailed. They represent a mix of excitatory and inhibitory actions, depending on the specific neuron releasing them, the location in the gut, and the type of receptor on the target cell. They mediate many of the specialized functions, things like sphincter relaxation, VIP, pain signaling, substance P, satiety signals, CCK, and coordinating complex reflex patterns. The details often get covered when discussing specific gut regions or functions.
Okay, so a very rich chemical language within the gut's own brain. Now let's talk more about those outside influences, the autonomic nervous system. How does it connect?
Right, the extrinsic control. The parasympathetic and sympathetic systems provide that crucial link between the gut and the rest of the body's state.
Let's start with the parasympathetic. What's its general effect?
The parasympathetic nervous system generally acts to increase the activity of the enteric nervous system. Think of it as the rest and digest system. It stimulates gut function. It's the accelerator pedal.
Where does this parasympathetic input come from?
It comes from two main divisions. There's the cranial division, primarily traveling through the massive vagus nerves. These originate in the brainstem.
The vagus nerves. They're really important for the gut, aren't they?
Hugely important. They provide extensive innervation to the esophagus, the stomach, the pancreas, and the entire small intestine, extending down into the first half of the large intestine. They are the major parasympathetic pathway for the upper and midgut.
And the second division.
That's the sacral division. It originates lower down in the spinal cord, from segments S2, S3, and S4. Fibers from here travel via the pelvic nerves.
Where do the pelvic nerves go?
They primarily supply the distal half of the large intestine, the descending colon, sigmoid colon, rectum, and the anal region. These nerves are particularly critical for controlling the powerful contractions involved in the defecation reflexes.
And when these parasympathetic nerves reach the gut wall, where do they connect? Directly to muscle.
Mostly not directly. A key point is that the final neurons in the parasympathetic chain, the postganglionic neurons, are located predominantly within the myenteric and submucosal plexuses of the ENS itself.
Ah, so the vagus or pelvic nerves signal to the ENS neurons, and then the ENS neurons signal to the muscle or glands.
Exactly. The extrinsic parasympathetic input primarily works by stimulating or modulating the activity of the gut's own intrinsic nervous system. So stimulating these parasympathetic nerves generally ramps up the entire ENS network they innervate, leading to enhanced gut motility and increased secretion.
Okay, parasympathetic speeds things up via the ENS. What about the sympathetic system? Opposite effect.
Generally, yes. Sympathetic stimulation usually has the opposite effect. It inhibits the activity of the gastrointestinal tract. It's the fight-or-flight response. Digestion is not a priority. It's the brake pedal.
Where does sympathetic input originate?
It originates in the spinal cord, typically from segments T5 down to L2.
And the pathway to the gut.
The initial fibers, the preganglionic fibers, travel out to synapse in large ganglia located outside the gut, like the celiac ganglion, superior mesenteric ganglion, and inferior mesenteric ganglion. The postganglionic neurons are in these ganglia, and their fibers then spread out to supply all parts of the GI tract from top to bottom. It's a very widespread distribution.
What neurotransmitter do they use?
They primarily secrete norepinephrine.
And how does norepinephrine inhibit the gut? Does it act directly on the muscle?
It has two main mechanisms, according to the source. There is a slight, direct inhibitory effect of norepinephrine on the main smooth muscle layers, longitudinal and circular. Interestingly, it actually excites the thin mucosal muscle, but that's a minor effect overall.
What's the main inhibitory mechanism then?
The major inhibitory effect of sympathetic stimulation comes from norepinephrine strongly inhibiting the neurons of the entire enteric nervous system.
So it shuts down the gut's own brain.
Pretty much. It suppresses the activity of both the myenteric and submucosal plexuses. The source really emphasized how powerful this is. Strong sympathetic stimulation can inhibit gut motor movements so completely that it can literally block the passage of food through the entire GI tract. It can bring things to a halt.
Wow. So a clear division. Parasympathetic stimulates via the ENS. Sympathetic inhibits mainly by suppressing the ENS. It's a balanced system.
A beautifully balanced control system. But for this control to work properly, both the ENS and the central nervous system need constant information about what's actually happening inside the gut lumen wall. That's where the afferent sensory nerve fibers come in. They provide the crucial feedback.
The gut's own senses.
Exactly. The gut is loaded with these sensory nerve endings and fibers. The cell bodies of these sensory neurons are located either within the ENS itself or in the dorsal root ganglia, which lie just outside the spinal cord.
What kinds of things do they sense?
They're tuned to detect several key conditions, as listed in the source. Physical or chemical irritation of the gut lining, mucosa, think toxins, very high acidity, rough particles. Also, excessive distension or stretching of the gut wall, like when it's overfilled with food or gas. And the presence of specific chemical substances in the gut contents, things like certain nutrients, digestive products, or molecules released by bacteria.
And when these nerves are activated, what happens?
The signals they generate can trigger various responses. Some signals stay local, activating reflex arcs entirely within the ENS to cause, say, increased mucus secretion to protect against an irritant, or maybe inhibiting movement if there's excessive distension.
But some signals go further out.
Yes. As we mentioned, many afferent fibers travel outside the gut wall to the perivitral ganglia, the spinal cord, or all the way to the brainstem. And it's worth highlighting the vagus nerves again here.
We usually think of them carrying signals to the gut.
Right, the parasympathetic motor signals. But the source points out that a staggering 80% of the nerve fibers within the vagus nerves are actually afferent, meaning sensory fibers carrying information from the gut up to the brainstem, particularly the medulla.
80%. So the vagus is mostly a sensory highway from the gut to the brain.
Overwhelmingly so. It allows the brainstem to have a constant, detailed update on the state of the upper and mid-GI tract, how stretched it is, what chemicals are present, whether there's inflammation. This information is then used to generate appropriate reflex responses, often sent back down via the efferent motor vagal fibers. It's a major component of that gut-brain communication axis.
Okay, so we have the intrinsic ENS, the extrinsic autonomic modulation, and this constant sensory feedback. This must create some complex gastrointestinal reflexes.
Absolutely. The interplay between these components results in various reflex loops that govern gut function. The source nicely categorizes these into three essential types based on where the signals are processed or integrated.
Type 1.
Type 1 reflexes are those integrated entirely within the gut wall enteric nervous system. These are purely local affairs.
So the ENS handles these itself, without bothering the brain or spinal cord.
Exactly. These reflexes allow the gut to respond rapidly and autonomously to local conditions. Examples include controlling much of the localized secretion of digestive juices in response to food presence, initiating the basic peristaltic contractions in response to stretch, regulating the mixing movements, segmentation, and mediating those local inhibitory effects, like the relaxation of muscle just ahead of a peristaltic wave. The ENS is remarkably self-sufficient for many basic gut operations.
Okay, local control within the wall. What's type 2?
Type 2 reflexes involve signals traveling from the gut up to the prevertebral sympathetic ganglia. Those ganglia, like the celiac and mesenteric, located outside the gut wall and then back down to the GI tract.
So these connect different parts of the gut.
Yes, these are crucial for coordinating activity between different, often distant regions of the GI tract. Because they route through sympathetic ganglia, they often involve inhibitory signals.
Can you give some examples?
Sure. A classic one is the gastrocolic reflex. When the stomach stretches after you eat a meal, signals travel up to the prevertebral ganglia, which then sends signals down to the colon, triggering mass movement, those large contractions that propel feces towards the rectum, often leading to the urge to defecate after eating.
The after-meal urge.
That's the one. Another key set are the enterogastric reflexes. Entero meaning intestine, gastric meaning stomach. Signals originate in the small intestine or colon, usually in response to distension or specific nutrients like fat or acid, travel to the prevertebral ganglia, and then send inhibitory signals back to the stomach, slowing down its motility and secretion.
Why slow the stomach?
To prevent the lower parts of the intestine from being overwhelmed, if the duodenum is already full of fat or acid, this reflex tells the stomach, hold on, slow down, give us time to process what we've got.
Makes sense. Any others?
The colonoidal reflex is another. Signals from the colon, especially when it's distended, travel via the ganglia to inhibit the emptying of contents from the very end of the small intestine, the ileum, into the colon. It protects the colon from getting overloaded too quickly.
Okay, so type 2 coordinates different gut regions via the sympathetic ganglia. What's type 3?
Type 3 reflexes are the ones that go all the way from the gut up to the spinal cord or the brainstem and then back down to the GI tract. These involve the central nervous system and allow for the most complex levels of control and integration with other bodily functions.
Like reflexes involving the brain itself.
Yes. For instance, reflexes controlling gastric motor and secretory activity that originate in the stomach and duodenum travel up the vagus nerve to the brainstem, medulla, are processed there, and then commands are sent back down the vagus nerve. These are often triggered by the presence and type of food in the upper gut.
What about pain?
Pain is a big one. Pain reflexes originating almost anywhere in the GI tract can travel to the spinal cord and trigger a general inhibition of the entire gastrointestinal tract. It's like the body saying, something's seriously wrong, shut down digestion for now.
And defecation, you mentioned that earlier.
Yes, the powerful defecation reflexes are a prime example of type 3. Distension of the rectum initiates signals that travel to the sacral spinal cord. The spinal cord integrates these signals and sends strong motor commands back via the pelvic nerves to the descending colon, sigmoid colon, and rectum, causing the powerful contractions needed to expel feces. Higher brain centers can, of course, exert voluntary control over the final step of this reflex.
Incredible. Local reflexes, regional coordination via ganglia, and central control via the spinal cord and brain. It's a truly layered system.
It really is, and we haven't even finished with the control systems yet. Besides the intricate nervous control, intrinsic and extrinsic, the gut's function is also heavily regulated by hormones. This is the third major pillar of control.
Hormones, chemical messengers in the blood, right?
Exactly. Unlike radic nerve signals traveling along specific wires, gastrointestinal hormones are chemical substances released by specialized endocrine cells within the gut mucosa directly into the local blood circulation specifically, the portal circulation that goes first to the liver.
So they travel everywhere in the blood.
They travel throughout the bloodstream, but they only act on target cells elsewhere in the gut, or sometimes other organs like the pancreas or gallbladder that possess specific receptors for that particular hormone. It's like sending a radio broadcast, but only radios tuned to the right frequency can receive the message.
And because they travel in the blood, their effects are slower but maybe longer lasting than nerve signals.
Generally, yes. Hormonal effects tend to develop more slowly, but persist as long as the hormone remains in circulation, even if all the nerve connections were cut. It provides a different timescale of regulation.
Our source mentions several key GI hormones. Do they affect movement?
Yes, many of them have significant effects on motility, although several also strongly influence secretion, as the source notes. We'll focus here on their described actions on motility.
Okay, who's first?
First up is gastrin. This hormone is secreted mainly by specialized cells called G cells, found mostly in the lower part of the stomach, the antrum, but also some in the upper small intestine, duodenum, jejunum.
What triggers gastrin release?
Things associated with eating a meal, primarily, the stomach stretching as it fills, the presence of protein breakdown products, peptides and amino acids in the stomach, and also stimulation by a specific peptide, gastrin-releasing peptide, released by vagus nerve endings during the thought or sight of food.
And what does gastrin do for motility?
Interestingly, while gastrin is most famous for being a powerful stimulator of gastric acid secretion and also promoting the growth of the stomach lining, the source indicates that its direct effects on large-scale gastrointestinal motility are usually considered less important than its secretory and growth effects. So, key hormone, but maybe less central for movement control compared to others.
Okay, next.
Next is cholecystokinin, universally known as CCK. This is secreted by eye cells, which are found in the lining of the duodenum, jejunum, and ileum, basically throughout the small intestine.
What makes the eye cells release CCK?
The primary stimulus is the presence of fat digestion products, specifically fatty acids and monoglycerides in the small intestine lumen. Protein products also stimulate it, but fat is the big trigger. It's the gut's main signal that fat has arrived and needs dealing with.
And what does CCK do for motility?
It has two really key actions listed. First, it causes strong contraction of the gallbladder. This squeezes stored bile out into the duodenum. Bile is absolutely essential for emulsifying fats, breaking them into smaller droplets so digestive enzymes can work on them effectively.
Makes sense. Fat arrives. Release the fat-digesting helper bile.
Exactly. Second, CCK also has a moderate inhibitory effect on stomach contraction. It slows down the rate at which the stomach empties its contents into the small intestine.
Why slow the stomach down?
Because digesting and absorbing fat is a relatively slow process. By inhibiting gastric emptying, CCK ensures that the small intestine receives the fatty meal at a manageable rate, giving it enough time to properly digest and absorb the fats before the next batch arrives. It prevents overload.
Clever. Anything else for CTK?
The source also notes its role in promoting satiety, the feeling of fullness. It seems CCK signals to the brain, likely via the vagus nerve, helping to inhibit appetite after a fatty meal.
Okay, CCK. Gallbladder squeeze, stomach break, feeling full. Got it. Who's next?
Secretin. Historically very important. It was actually the very first substance identified as a hormone over a century ago. It's secreted by S cells found mainly in the duodenal mucosa.
What triggers secretin?
Its main stimulus is acidic gastric juice entering the duodenum from the stomach. When the pH in the duodenum drops too low because of stomach acid, S cells release secretin. It's basically the acid alarm.
And its effect on motility.
The source states that secretin has only a mild effect on motility of the GI tract. Its primary and much more dramatic role is stimulating the pancreas to secrete a large volume of fluid rich in bicarbonate.
Bicarbonate to neutralize the acid.
Precisely. Secretin's main job is to protect the duodenal lining from acid damage and to raise the pH back towards neutral, which is the optimal environment for the digestive enzymes that work in the small intestine. Motility effects are secondary.
Okay, another one.
Yes, glucose-dependent insulinotropic peptide, or GIP. This one used to be called gastric inhibitory peptide, which gives a clue to one of its actions. It's secreted by K cells in the mucosa of the upper small intestine, duodenum and jejunum.
What stimulates GIP release?
Mainly the presence of fatty acids and amino acids in the small intestine lumen. Carbohydrates stimulate it too, but less strongly.
And its motility effect, the gastric inhibitory part.
Right. GIP does have a mild effect in decreasing the motor activity of the stomach. So, like CCK, it contributes to slowing down gastric emptying, particularly when the upper small intestine is already processing fats and proteins. Again, it helps match the delivery rate from the stomach to the processing capacity of the intestine.
But its main job is hinted at by the new name, insulinotropic.
Exactly. GIP's most crucial recognized function now is its powerful effect in stimulating insulin secretion from the pancreas, especially when glucose is also present in the gut lumen. It's a key incretin hormone, signaling the pancreas to prepare for the glucose load about to be absorbed from the meal. It links gut digestion directly to blood sugar regulation.
Fascinating connection. One more.
Last one mentioned here is motilin. This is secreted by M cells located in the stomach, antrum, and the upper duodenum.
What triggers motilin? Food.
Actually, no. Unlike the others, motilin secretion isn't primarily stimulated by food ingestion. It's secreted cyclically, primarily during fasting, in the periods between meals.
Fasting. What does it do then?
Its only known function highlighted in this source is to increase gastrointestinal motility during that fasting state.
Why increase motility when there's no food?
It's thought to be part of a gut housekeeping process. Motilin is released in cycles roughly every 90 minutes during fasting. This triggers waves of strong propagating contractions that sweep all the way from the stomach down through the small intestine. These waves are called the interdigestive myoelectric complexes, or sometimes the migrating motor complex, MMC.
Housekeeping ways.
Yeah, the idea is they sweep out any residual undigested food particles, slut off cells, mucus, and importantly bacteria from the stomach and small intestine, moving them down into the large intestine. This helps keep the upper gut relatively clean and prevents bacteria from overgrowing there. Once you eat, motilin secretion is inhibited and the gut switches back to the fed pattern of segmentation and slower peristalsis for digestion and absorption.
So motilin is the fasting street sweeper.
That's a good way to think of it. So you see how these hormones act as chemical signals, released in response to specific conditions: food presence, fat, acid, fasting to fine-tune motility and secretion, working alongside the nervous system to orchestrate the whole process.
Okay, we've covered the muscle, the electricity, the nerves, intrinsic and extrinsic, and the hormones. Let's bring it back to the actual results, the functional movements that make digestion happen.
Right, the mechanical actions. Our source boils these down to two fundamental types: propulsive movements, which obviously move the food forward along the tract, and mixing movements, which turn the contents to ensure thorough blending with digestive juices and good contact with the absorptive lining.
Propulsion first. What's the main mechanism?
The basic propulsive movement throughout most of the GI tract is peristalsis. We touched on this earlier. The source uses a great analogy. Imagine putting your fingers around a soft-filled tube, squeezing down, and then sliding that constriction forward along the tube. Everything ahead of your fingers gets pushed forward.
A moving ring of contraction.
Exactly. It's an inherent property of these syncytial smooth muscle tubes. If you stimulate a point, it tends to trigger a ring of contraction in the circular muscle that then propagates along the tube. We see similar movements in other body tubes like the ureters or bile ducts.
What usually triggers peristalsis in the gut?
The most common and effective stimulus is simply distension, the gut wall being stretched by the presence of a bolus of food or accumulated contents. This stretching activates the sensory nerves in the wall, which then signal the enteric nervous system, particularly the myenteric plexus.
And the ENS then triggers the contraction.
Yes, it triggers a coordinated response. A ring of circular muscle contraction forms a few centimeters behind the distended point on the orid side towards the mouth, while simultaneously the segment of gut ahead of the bolus on the anal side relaxes. Then the contractile ring moves forward, typically for 5 to 10 centimeters, pushing the bolus into the relaxed segment ahead.
Does anything else trigger it besides stretch?
Yes, chemical or physical irritation of the gut lining can also initiate peristalsis, as can strong signals from the parasympathetic nervous system.
You mentioned the myenteric plexus. How important is it for this?
The source is very clear. It's absolutely essential. Peristalsis is either very weak or completely absent in parts of the gut where the myenteric plexus hasn't developed properly, a congenital condition, or if its function is blocked, for instance, by drugs like atropine that paralyze the cholinergic nerves within the plexus.
So muscle needs the nerve plexus to do peristalsis properly.
For effectual peristalsis, yes. The coordinated pattern of contraction behind and relaxation ahead requires an active, functioning myenteric plexus orchestrating the muscle layers.
And this wave always goes one way, towards the exit.
That's the normal pattern. While stimulation could theoretically start a wave going backwards orally, those waves tend to die out very quickly. The waves moving towards the anus, however, propagate much more effectively and consistently. This unidirectional preference is often called the law of the gut.
Why does it prefer to go towards the anus?
The probable reason, according to the source, is that the myenteric plexus itself is functionally polarized in the anal direction. The circuitry is inherently biased to conduct signals more readily that way.
And you mentioned relaxation ahead of the wave.
Yes, that's a critical part of the pattern. As the contractile ring forms behind the bolus and starts moving forward anally, the gut segment downstream or ahead of the bolus simultaneously undergoes receptive relaxation.
Why is that relaxation important?
It dramatically lowers the resistance ahead of the advancing bolus. It opens the way, making it much easier for the contractile wave to push the contents forward efficiently. Without receptive relaxation, the wave would just be pushing against a constricted or non-relaxed tube, which is much harder work.
So the whole package contraction behind, relaxation ahead, moving towards the anus, that's the key.
That coordinated pattern is the key. It's formally called the myenteric reflex, or the peristaltic reflex. And when you combine this organized reflex with the inherent tendency for the movement to proceed towards the anus, that's what the source refers to as the law of the gut. It's the fundamental mechanism for orderly propulsion through the system.
Okay, that covers moving things along. What about mixing?
Mixing is also crucial, especially in the small intestine where digestion and absorption are happening intensely. How it's achieved can vary.
Can peristalsis do mixing too?
Sometimes, yes. If a peristaltic wave runs into resistance, like a closed sphincter or a segment already full of contents, the forward propulsion gets blocked. The wave then just churns the contents back and forth within that segment, effectively mixing them.
But is there a movement specifically for mixing?
Yes. Particularly in the small intestine, the primary mixing movements are called segmentation contractions.
Segmentation, what does that look like?
Imagine the intestine getting tinged off by localized, intermittent constrictions of the circular muscle that appear every few centimeters along its length. It divides the gut temporarily into multiple small segments.
Right, sausage links?
Kind of like that, yes. These constrictions only last for a short time, maybe 5 to 30 seconds. Then, crucially, as one set of constrictions relaxes, a new set appears at different points, usually halfway between the previous constrictions.
So it keeps dividing the contents in new ways.
Exactly. This pattern of dividing, relaxing, and redividing at new locations has the effect of chopping and shearing the intestinal contents over and over again. It thoroughly kneads the food, mixing it intimately with the digestive juices, bile, pancreatic enzymes, intestinal secretions.
And it must help absorption too.
Definitely. By constantly sloshing the mixture around, segmentation ensures that all the digested nutrients are repeatedly brought into close contact with the absorptive mucosal surface. So peristalsis, propulsion, segmentation mixing. 2 key motor patterns.
All this activity, the muscle contracting, the glands secreting, the cells absorbing, must need a huge amount of energy and blood flow.
An enormous amount. Which brings us to the gastrointestinal blood flow, often discussed as part of the larger splanchnic circulation.
Splanchnic, what does that include?
The splanchnic circulation refers to the blood flow through the gut itself, stomach, intestines, plus the blood flow through the associated organs, the spleen, the pancreas, and very importantly, the liver.
And this circulation is set up differently, isn't it? Something about the liver.
Yes, it has a unique and critical design, clearly shown in the source diagrams. All the venous blood draining from the capillaries of the gut, stomach, small and large intestines, the spleen, and the pancreas doesn't go directly back towards the heart like blood from your legs or arms would.
Where does it go?
It all collects into one large vein called the portal vein.
The portal vein.
Now, this portal vein then carries all of that blood. Blood, now potentially rich in absorbed nutrients from the gut, blood that's passed through the spleen, which filters blood and plays immune roles, and blood from the pancreas, carrying hormones like insulin and glucagon, directly into the liver.
So the liver gets first dibs on everything coming from the gut.
Essentially, yes. Inside the liver, the portal vein branches out, and the blood flows through a special network of wide, leaky capillaries called the liver sinusoids. Only after passing through these sinusoids does the blood collect into the hepatic veins, which then finally drain into the inferior vena cava, the main vein returning blood to the heart.
Why this detour through the liver? What happens in the sinusoids?
Two absolutely critical things. First, the sinusoids are lined with specialized immune cells, a type of macrophage called reticuloendothelial cells, or Kupffer cells in the liver. These act like vigilant gatekeepers. They phagocytose, meaning they engulf and destroy bacteria and any other particulate matter that might have been absorbed from the gut, along with the nutrients.
So it's a filter, preventing gut bacteria from getting into the main bloodstream.
Exactly. It's a vital protective filter, preventing systemic infections originating from the gut. Second, the liver cells themselves, hepatocytes, that line the sinusoids get the first chance to process the absorbed nutrients.
What kind of processing?
For water-soluble nutrients like carbohydrates, sugars, and amino acids from proteins, which are absorbed into the gut capillaries and travel via the portal vein, the liver cells absorb and temporarily store a large fraction, the source says maybe 50% to 75% of this incoming load. They perform crucial intermediary metabolism, converting sugars, storing glucose as glycogen, processing amino acids, detoxifying substances, etc., before releasing the modified nutrients into the general circulation in a more controlled way.
So the liver acts as a buffer and processing plant for nutrients. What about fats? Do they go through the portal vein too?
Mostly no. This is a key difference. Most digested fats are absorbed differently, packaged into particles called chylomicrons, and taken up by the lymphatic capillaries, lacteals, in the gut villi, not the blood capillaries.
Lymphatics, where do they go?
The lymphatic system is a separate network of vessels. The lymph fluid containing the fats eventually collects and drains into the bloodstream via the thoracic duct, which empties into a large vein near the heart. So, absorbed fats largely bypass the initial passage through the liver via the portal vein.
Interesting distinction. Okay, let's look at the blood vessels supplying the gut itself. Where does the arterial blood come from?
The major arteries, as shown in the sources diagram, are the celiac artery, supplying the stomach, liver, spleen, pancreas, upper duodenum, and the superior and inferior mesenteric arteries, supplying the rest of the small and large intestines.
Do they go straight into the gut wall?
Not quite. They first branch extensively in the mesentery, that sheet of tissue anchoring the intestines, forming interconnected arches. Smaller arteries then arise from these arches, circle around the gut tube, and then penetrate inwards into the gut wall.
And inside the wall.
Once inside, they spread out, sending branches along the muscle bundles to supply the longitudinal and circular layers. They also send branches deeper into the submucosa and then up into the mucosa, forming dense capillary networks. These mucosal capillaries are especially elaborate within the intestinal villi, those finger-like projections where most absorption happens.
What's the setup inside a villus?
The source shows a great illustration. Typically, a small arteriole runs up the center of each villus. It then branches into a dense looping network of capillaries located just underneath the absorptive epithelial cells at the surface. These capillaries then collect into a venule that runs back down the center of the villus, carrying away the absorbed nutrients, except the fats and lymph.
And can the gut control blood flow into these villi?
Yes, the arterioles feeding the villi are quite muscular and can actively constrict or dilate, allowing for precise control over how much blood flows through the villus capillary network at any given time.
Which leads to the question, how is blood flow controlled? Does it just stay constant?
Definitely not. One of the most striking features is that, under normal conditions, blood flow in any specific area or layer of the gut wall is directly related to the level of local activity in that area.
More activity, more blood flow.
Exactly. It's a beautiful example of local metabolic control. If a segment of muscle is contracting vigorously for mixing, blood flow to that muscle layer increases. If glands in the mucosa are actively secreting digestive juices, blood flow to the mucosa increases.
And during absorption.
That's when you see the biggest jump. The source says blood flow in the villi and submucosa can increase by as much as 8-fold during periods of active nutrient absorption.
8 times, that's huge.
It is. So overall, after you eat a meal, when a motor activity, secretion, and absorption are all ramped up, the total blood flow to the splanchnic region increases dramatically. This increased flow, called postprandial hyperemia, typically lasts for 2 to 4 hours, gradually returning to resting levels as digestion and absorption wind down.
What causes this huge increase in blood flow? How does the gut signal the vessels to open up?
The precise mechanisms are still being fully unraveled, but the source discusses several likely contributing factors. It's probably a combination of things.
Like what?
One major factor is the release of various vasodilator substances from the gut mucosa itself during digestion. Interestingly, many of these appear to be the same peptide hormones we discussed earlier that control motility and secretion, things like cholecystokinin, CCK, vasoactive intestinal peptide, VIP, gastrin, and secretin.
So the hormones that trigger gut activity also help increase its blood supply.
It makes perfect physiological sense, doesn't it? Coordinate the function with the fuel delivery. Another group of vasodilators involved are the kinins, specifically kalitin and bradykinin. These are powerhouse vasodilators released into the gut wall by some glands during active secretion, contributing to the increased mucosal blood flow.
What about metabolic factors? Does the gut's hard work itself signal for more blood?
Yes, that seems very important. As the gut tissues become more metabolically active, burning more fuel, using more oxygen, the local oxygen concentration likely decreases. Lower oxygen levels in the tissue are themselves a potent stimulus for vasodilation, potentially increasing flow by 50–100% just from that effect.
Low oxygen triggers more flow to deliver more oxygen.
Exactly. And related to that, the decreased oxygen and increased metabolic activity can lead to a significant increase, maybe up to 4-fold, in the local concentration of adenosine. Adenosine is a well-known and powerful vasodilator in many tissues, and it's thought to be responsible for a substantial portion of the increased blood flow seen during gut activity.
So it's a mix. Hormones, kinins, low oxygen, and adenosine, all telling the blood vessels to relax and let more blood through when the gut is busy.
That's the current understanding. It's likely a complex interplay of these and possibly other factors, all working together to precisely match blood flow to the metabolic demands of digestion and absorption.
Now, you mentioned the blood vessels inside the villi earlier. Is there anything special about the flow pattern there?
There is, and it's fascinating, but also creates a potential vulnerability. It's the countercurrent blood flow arrangement within the villi.
Countercurrent, what does that mean?
If you look closely at the diagram, like figure 63-8 in the source, you see the small arteriole carrying oxygenated blood up into the villus runs right alongside the venule carrying deoxygenated blood down and out of the villus. They are in very close proximity and the blood is flowing in opposite directions. That's a countercurrent setup.
Okay, artery going up, vein coming down, right next to each other. What's the consequence?
Because they're so close and flow is in opposite directions, a significant amount of oxygen, the source estimates, as much as 80% can diffuse directly from the high oxygen arterial blood across the small gap into the low oxygen venous blood, effectively bypassing the capillary network at the very tip of the villus.
80% bypasses the tip. That sounds inefficient. Isn't the tip where absorption happens?
It is where a lot of absorption happens. And under normal conditions, when blood flow is high after a meal, this shunting isn't usually a problem. Enough oxygen still reaches the tip capillaries to meet their metabolic needs for active transport and absorption. The source compares the setup to the countercurrent mechanism in the kidney's vasorecta, although the functional implication here is oxygen shunting.
But you said it creates a vulnerability. When would this be a problem?
It becomes critically important in situations of low overall blood flow to the gut, such as during circulatory shock, for example, from severe blood loss or dehydration.
What happens in shock?
In shock, the total blood flow entering the villus arteriole is already drastically reduced. When you combine that low inflow with the 80% oxygen shunt directly to the venule, very little oxygen, perhaps critically little, actually makes it all the way to the capillaries at the villus tip.
So the very tip becomes starved of oxygen.
Exactly. The oxygen deficit at the tip can become so severe that the cells there suffer ischemic injury damage due to lack of oxygen and can die and disintegrate. This can lead to the blunting or even complete destruction of the villus tip, or sometimes the entire villus.
Wow, so this countercurrent flow makes the villi tips especially sensitive to poor circulation.
It's believed to be a major reason why the intestinal lining, particularly the villi, is so susceptible to damage in shock states or other diseases causing severe gut ischemia. This damage, in turn, dramatically reduces the absorptive surface area of the intestine, which can severely worsen the patient's condition. It's a fascinating trade-off in design.
Okay, so local factors are huge for controlling blood flow.
Mhm.
Does the nervous system play a direct role, too?
It does, although its role is a bit different from the fine-tuning provided by local metabolites. Let's look at the autonomic inputs.
Parasympathetic first. Does rest and digest increase blood flow?
Parasympathetic stimulation, particularly to the stomach and lower colon where its innervation is dense, does correlate with increased local blood flow. However, the source suggests this is likely a secondary effect.
Secondary to what?
Secondary to the increased glandular activity. Parasympathetic nerves strongly stimulate secretion from glands in those areas. This increased metabolic activity of the glands likely triggers local vasodilation through the mechanisms we just discussed, low O2, adenosine, etc. So the increased flow might be driven more by the gland's needs than by a direct effect of the nerves on the blood vessels themselves.
Okay, so parasympathetic indirectly boosts flow by boosting activity. What about sympathetic, fight or flight?
Sympathetic stimulation has a very potent and direct effect. It causes intense vasoconstriction of the arterioles throughout essentially the entire GI tract, including the mesentery and spleen. This acts to greatly decrease blood flow to these organs,
Why would the body want to cut off blood flow to the gut?
primarily to redirect that blood elsewhere during emergencies or periods of high demand by other tissues. But there's an interesting twist called autoregulatory escape.
Escape. What escapes?
The blood flow escapes from the sympathetic constriction. What happens is, even if the strong sympathetic stimulation continues, after a few minutes, the blood flow to the gut often returns towards or close to normal levels.
How? If the sympathetic nerves are still firing.
It's because those powerful local metabolic vasodilator mechanisms kick in. The initial sympathetic vasoconstriction causes temporary ischemia, reduced blood flow, which triggers the release of adenosine, kinins, etc., and lowers oxygen. These local vasodilator signals become strong enough to override the sympathetic constrictor signal, forcing the arterioles to relax again to restore necessary blood flow to the working gut tissues.
So the local metabolic needs can eventually win out over the nervous command to constrict.
To a large extent, yes. It ensures the gut doesn't suffer prolonged damaging ischemia just because the sympathetic system is active. However, the initial vasoconstriction is still very important physiologically.
Why is that initial shutdown valuable?
Its major value, as the source emphasizes, is that it allows the body to temporarily shut off or significantly reduce blood flow to the entire splanchnic region when that blood is urgently needed elsewhere.
Like during heavy exercise.
Exactly. During intense exercise, your skeletal muscles and heart need a massive increase in blood flow. The sympathetic system constricts splanchnic vessels, diverting blood away from the gut, which isn't a priority during a sprint, towards the working muscles.
Or in shock.
Yes, critically important in circulatory shock. When blood volume is low, sympathetic constriction of the gut vessels helps maintain blood pressure and perfusion to the absolute vital organs, the brain and the heart. This effect can persist for many hours if needed, essentially sacrificing gut perfusion temporarily to ensure survival.
Does sympathetic stimulation affect the veins too, not just the arteries?
It does. Sympathetic nerves also cause strong vasoconstriction of the large volume intestinal and mesenteric veins.
Why constrict the veins?
These splanchnic veins normally hold a considerable amount of blood. They act as a blood reservoir. When the sympathetic system constricts them, it decreases their capacity and squeezes that stored blood out into the general circulation.
How much blood?
The source estimates this can displace 200 to 400 milliliters of blood. That might not sound huge, but in a situation like hemorrhagic shock, where every milliliter counts, adding that volume back into active circulation can provide crucial support to maintain blood pressure and overall circulatory function. It's another way the splanchnic circulation helps the body cope with emergencies.
So putting it all together, this deep dive really shows the alimentary tract isn't just some simple plumbing.
Not at all. It's a truly sophisticated, dynamic, and highly regulated system. A masterpiece of biological engineering, really.
We've seen its complex electrical behavior, the slow waves setting the rhythm, the spike potentials triggering contraction through calcium influx.
We've explored its own intrinsic nervous system, the ENS, this second brain, with incredible complexity, capable of local reflexes and coordinating movement over long distances via things like the myenteric reflex and that fundamental law of the gut.
And how that intrinsic system is constantly modulated by the extrinsic autonomic nerves, the parasympathetic accelerator, and the powerful sympathetic brake.
Plus, the whole layer of hormonal control, with chemical messengers released in response to food cues or fasting states, fine-tuning motility and secretion, often working hand in glove with the nervous signals.
We saw how movement isn't just pushing, but this coordinated dance of peristalsis for propulsion, guided by receptive relaxation and segmentation for incredibly thorough mixing.
And underpinning it all, the vital splanchnic circulation, not just supplying the energy for all this activity with blood flow precisely matched to local needs, but also playing that critical protective role by filtering blood through the liver.
And acting as that crucial emergency blood reservoir, thanks to sympathetic control, even though that countercurrent flow in the villi creates a weak spot in extreme conditions.
It's just an amazing integration of muscle physiology, complex neural networks, hormonal signaling, and specialized vascular design. Local controls mesh seamlessly with systemic commands.
It really is awe-inspiring. And, thinking about how interconnected everything is, the nerves, the muscles, the blood flow, all responding to the local environment, it raises a further question, doesn't it?
What are you thinking?
Well, our source focused on the gut wall and its controls, but we know the gut lumen itself is teeming with life, the gut microbiome, trillions of bacteria. We didn't cover their influence from this material, but given how sensitive the ENS is to chemical signals and how blood flow responds to metabolic changes.
You're wondering how those microbes living right there in the lumen might be interacting with or even modulating all these systems we've just discussed.
Exactly. How might the microbiome's own signaling molecules or metabolic byproducts influence the enteric nerves, the hormone releases, maybe even the muscle excitability or the local blood flow control we've detailed today? It wasn't in our source, but it feels like a whole other layer of complexity interacting with this machinery, something to definitely mull over.